Dear students, I am inviting you all to our Sri Chaitanya's a new approach for the consolidation of each and every chapter. In fact, it is an unique method of consolidation. Why? Because we are inching towards the conclusion of our preparation. Few days later, we are going to have the examination. So, we are in the final step of revision. In this stage, reading, revising, it is naturally happening. But during this reading process, revision process, some of the points may be neglected. Some of the points may not be registered properly in the minds of the student. Some of the areas which are very vital, they need some stressful reading that may escape. So to show that areas, we are providing one bird's eye view on every chapter. That unique and new approach of Sri Chaitanya is chapter at a glance. Today's topic is molecular basis of inheritance. We know that in our plus 2 textbook, we have genetics part. Genetics, we have 2 chapters. Principles of inheritance and variation, first one. After that, molecular basis of inheritance. And principles of inheritance and variation, where we read about Mendel's laws, monohybrid cross, dihybrid cross, test cross, back cross, etc., etc. After that, we will come to the human genetics part, genetic disorders, pedigree analysis, and chromosomal syndromes. And those are the highlights of the first chapter. After that, when you come back to our today's chapter, molecular basis of inheritance, and this chapter begins with the structure of DNA, DNA replication, then genetic code, then crossing over, etc., etc. And that part, the beginning part of molecular basis of inheritance, our botany sirs will show the chapter at a glance in their videos. But as far as zoology part is concerned, in this chapter, we have 2 subtopics. Subtopic number 1, Human Genome Project. Number 2, DNA Fingerprinting. So, I am showing that areas where the points to be clarified more, where a stressful reading is required, where some data is there that should be revised many times here. So definitely the video will help you for perfect consolidation to reach the answer of each and every question. So this is our topic, molecular basis of inheritance. 2 subtopics, one is called as the Human Genome Project, second one is DNA Fingerprinting. Now, first of all, look at here. This is the average number of questions in NEET per year. When now taken the average, generally one question will appear from these 2 subtopics, the so-called Human Genome Project and DNA Fingerprinting, generally. Then, how they are appeared, we have taken 3 years here, 2025, 2024, 2023. Now, see this. I am giving the year-wise data. 2025, we have received just 1 question. 2024, no question. 2023, 1 question. The reason for this molecular basis of inheritance, early part, whatever botany lectures are dealing, and this part together can be considered as one chapter. In this chapter, generally 2 to 3 questions will appear in every paper. Some will 1 or 2 in the botany paper, 1 or 2 in zoology paper. So that's why definitely 1 question is compulsory. But when I was seeing the previous history, 2023, 2024, 2025, most of the questions were asked multiple choice questions only. But don't believe this. Why? Because we have taken 3 years only. 2025 multiple choice question, 24 no question, 2023 1 question. These are multiple choice questions, but when you look into the previous years, where you can see statement wise questions are there, and two-column matching, data-wise questions were also there. So that's why it's a guidance. But these two subtopics of molecular base of inheritance, what we the zoology lectures are dealing, in which this is the data here. Now, see this 2025. 2025, that is last year, where we got a question, which chromosome in the human genome has the highest number of genes? You know it well, chromosome 1 here. So simple question, straight question, and the options are having data, be careful here. After that, already I mentioned that in 2024, no question was asked. No question was asked in the beginning part of the chapter, and 2 questions were appeared. So minimum 2 to 3 questions, maximum 4 questions I have seen in this molecular base of inheritance. That's it. But in my zoology part, this is 2023, we have received one more question, that is expressed sequence tags. ESTs refers to as actually all genes that are expressed as RNA. Actually in Human Genome Project, 2 methods were adopted. Those are 2 major approaches. One of the approaches is expressed sequence tags, in which all genes are identified that are expressed as RNA. So these are the questions here. Now you can have an idea. A lot of data is there. Read it, revise it repeatedly, make them familiar in your mind. Methodologies, 2 methods are there. I will just discuss now. I will show the NCERT book also, and you have to see that also. Some slight logical areas are present in DNA fingerprinting, and focus on that area. So get ready. Now I am going to show you the NCERT book. Yes, I am showing you our NCERT book. It is +2 textbook here. And the chapter name is Molecular Base of Inheritance. This is subtopic 5.9 Human Genome Project. What exactly the project is, you know it well. The sequencing, the complete genome of man, of a single cell, is Human Genome Project. Earlier we thought that this was almost all impossible because of increasement of science and technology. In the field of experimental biology, new molecular techniques were invented, followed by rapid growth occurred in computers. And in addition to these 2, the third point is added, that is Frederick Sanger. You know it well. That gentleman discovered automated DNA sequencing method, all come under this area, genetic engineering techniques. So new techniques were developed in the field of experimental biology, and rapid growth occurred in computers. And number 3, the automated DNA sequencing method that was discovered, invented by the Frederick Sanger. That's why it became absolutely possible. Absolutely possible, that's it. So here, the project of sequencing human genome was launched in the year 1990. This year is important always. When did the project was started? 1990. When it was completed? 2003. 2003. Total duration, the project 13 years. This data should be remembered always. And it was launched, beginning in 1990, completed 2003, and total 13 years. Of course, 2006, where the first chromosome was sequenced. That memory data you have to see here. Then actually, when I have seen this human genome, it is the data is given, you see that. Here, human genome is said to have this 3 into 10 to the power of 9 base pairs. The total 3 into 10 to the power of 9 base pairs is an estimated area. In that area, suppose if you want to sequence it, the cost of one base pair is $3. The total nucleotide base pairs are 3 into 10 to the power of 9. And the cost to sequence one base pair, $3. Then the estimated cost of the project would be approximately $9 billion. So 3 informations you have to remember. 3 into 10 to the power of 9 base pairs, the total human genome, and the cost to sequence one genome, one base pair, it is $3. The total cost of the project is $9 billion. $9 billion, that's it. Then later, And if you want to print it on a books, on paper, and the data is given, you see here. Very, very important. Here, each page of the book contained 1000 letters. And each book contained 1000 pages. And like that, 3300 books are required to print the human genome. Total number of books are required, 3300. Each book should have 1000 pages. Each page should have 1000 letters. I repeat once again, one page consists of 1000 letters. Such a type of 1000 pages are making a book. Such a kind of 3300 books are required here to store it. Very important. Now the goals are given. See the goals here, very, very important. 6 goals are given. Simply they are asking which of the following is the goal of a Human Genome Project. First of all, identify all approximately 20 to 25,000 genes in human DNA. That estimated. Generally the genes may be the 20,000 minimum, 25,000 we have to identify all. Then after that, determine the sequence of 3 billion chemical base pairs here. The first goal is the estimated genes 20,000 to 25,000. They have to identify it. Then second goal, and determine the sequences of all 3 billion nucleotide base pairs. That is the second one. And coming to the third one, store these information. It may be 3 billion base pairs or genes in databases. Store them in databases. Not only that, improve the tools for data analysis. Data analysis is there, and to improve the data analysis, you have to develop the computer programs here. Computer programs, so that is important. So first of all, they have to determine the sequence of all 3 billion nucleotide base pairs. Later, they have to identify 20,000 to 25,000 protein coding genes. That's the second one. Third one, store them in databases. Then providing tools for data analysis. This is the important point. After that, the related information should be transferred to the private agencies. Why? Because the project may have some defectives and that defect should be rectified. That means you have to transfer, see this point here, transfer of related technologies to the other sectors. It may be private sector or public sector, that's not the point here, and accuracy is aimed, that's the main reason in this area. After that, addressing ethical, legal, social issues. Ethically, some are believed to be correct in India, the same point is not followed by other countries. Socially, we have some conditions in India, but in European countries, in the United States of America and somewhere else, and they believe other customs here. Why all these are happening? So they have to address the ethical issues, legal issues, social issues. And these are the points here. Once again, identification of 20 to 25,000 protein coding genes, then the sequencing of all 3 billion nucleotide base pairs, then storing them in databases, providing tools for data analysis, then transfer this information to the other agencies. They will cross-check. If any correction is needed, they will make it. The aim is accuracy is important here. After that, addressing the ethical, legal, social issues. And have a think on this area. Then after that, already I told you that Human Genome Project was 13 years old project. 13 years. Started in the year of 1990, completed in 2003. 2003, that means 13 years project here. In which the previous question here you can see, it was coordinated by US Department of Energy, National Institute of Health. US Department of Energy and the National Institute of Health coordinated this program. In the early years, this is also previous question, in the early years, the Wellcome Trust of UK, see, the Wellcome Trust of UK became the major partner. That means US Department of Energy, National Institute of Health, and they have coordinated. That's one question. Another question, in the early years, the major partner was Wellcome Trust of UK, that's it. Then already I mentioned 2003, that's the year of completion, and that's it. Then actually what is the use here? the use is nothing but not only understanding about the entire human genome, here new ways to diagnose, treat, and someday present thousands of people are there suffering from the genetic disorders, and how to diagnose the disease, how to treat the disease, how to provide comfort to the people. This is the main idea. Now, I am showing a flow chart here. You see that. This is. The total highlights will come. First of all, let us see. Human Genome Project was a mega project. It was a large project. Because a lot of funding is required, you have seen a lot of money is required, it was a mega project. Not only money here, a lot of technology support is required. Not only that, a lot of time is also required. So that's the point here, and all highlights I brought here. Human genome is said to have approximately 3 into 10 to the power of 9 base pairs. Then the estimated cost of each base pair, $3 here, and the total project cost $9 billion. So 3 into 10 to the power of 9 base pairs, that is human genome, and cost of one base pair, $3, then $9 billion, and it is the total cost of the project here. Now coming to the next one, if we obtain sequences were to be stored in typed form in books, already we mentioned, but you take them outside and seeing 1 or 2 times, all points will register in your mind. That's the idea here. So if you want to type it or print it on a book, what is required here? If each page of the book contained 1000 letters, each book contained 1000 pages, such a type of 3300 books are required. Just to print the human genome, a single cell human genome, 3300 books are required. Each book should consist of 1000 pages. Each page should consist of 1000 letters. These are the highlights of this area. Now come to let us continue our NCERT book here. Now mark these discoveries. Now you see that here, in addition to the human genome, some other non-human models, organisms are there, which are also sequenced here, like bacteria, Escherichia coli, yeast cells, then Caenorhabditis elegans, Drosophila, etc. here. Here bacteria means Escherichia coli, you know it well. Then here Caenorhabditis elegans, most important point this is, it is a free-living non-pathogenic nematode. Very important here. It is a nematode, comes under Aschelminthes, and the Caenorhabditis elegans is also mentioned here. Then Drosophila melanogaster, the fruit fly, you know it well. Then plants like rice and Arabidopsis thaliana. Arabidopsis thaliana is a medicinal plant. That Arabidopsis thaliana, then rice, Oryza sativa, Drosophila melanogaster, an insect here. And most important one, see that, Caenorhabditis elegans, most important. Then the bacteria and yeast, you can go here. Then after that, how to sequence here, we have some methodologies. What are the methods employed here? Methods means what is the target? Very simple. 3 billion nucleotide base pairs are estimated, all must be sequenced. Second one, all genes should be identified. Generally, it may be 20,000 to 25,000. And you have to identify it. But what are the methods employed by the people? Different methods we have seen. But the two major approaches are there. One is expressed sequence tags. See this. Expressed sequence tags. Here, all genes, just now I have shown you a previous question. What are expressed sequence tags? The method in which all genes that are identified as RNA. That means the gene is expressed as RNA and these are identified, in which only genes are identified. Then coming to the another one, second method you see here, this second method. It's a blind approach. In the first expressed sequence tags, only we can identify genes. So some of the people gone for that, to identify the genes, to pick them out, to record them. And that is first method. But that first method can fulfill only one goal. But the entire genome should be sequenced. That's why second method, it's a blind approach. Total DNA is taken, it is broken into pieces, separated into coding sequence, non-coding. Both are separated. Later these are arranged. This is called as sequence annotation. So some adopted expressed sequence tags. Here only genes were identified that are expressed as RNA. But second method is a blind approach, but this approach is giving entire genes information, non-information. Very, very important here. Then after that, the commonly used hosts. What are the hosts used here? Bacteria and yeast are used here. In addition to that, the bacterial artificial chromosome, and yeast artificial chromosomes are used as vectors. Then here the fragments were sequenced using automated DNA sequencers. Here the gentleman who invented the method, automated DNA sequencing method, by using that, the Friedrich Sanger invented that method. Here you take a long fragment of DNA and apply the DNA to the automated DNA sequencing method. You will get the sequence within a short time in a perfect, accurate manner. That's it. Then this Friedrich Sanger is also famous for one more invention. This is the point here. That is determination of amino acid sequence in proteins. Two landmarks in this field. It was given by Friedrich Sanger. One is invention of automated DNA sequencing method. Second one, he also known for the determination of amino acid sequence in proteins. Yeah, that's it. Then after that, the specialized computer-based programs were developed. Because here in this field, normal computers were not used. They have used supercomputers. So that's why by taking the support of supercomputers, they have completed that. Then this is very, very important. Be careful. The project was started in the year 1990. By taking 13 years, it was completed in 2003. But this is very, very important. You see that in the month of May 2006. Very, very important. And the sequence of chromosome 1 was completed. Started in 1990 to 2003, the International Consortium of the Human Genome Project declared that the project was completed with an accuracy of 99.99%. This is most important point. In the month of May 2006, the chromosome 1, on which you have maximum number of genes are present, and that was completed. Then one more point you have to mention that the haploid set of chromosomes, the DNA present on haploid set of chromosomes, that constitutes the genome, not correct? And 24 chromosomes you have to take. That means 22 autosomes, 1 X chromosome and Y chromosome that constitute our genome. So this is the kind. That's our Human Genome Project. Just I'm giving an idea. The methodology is slight confusion may be there. That's why I've mentioned a flow chart here. See this flow chart. Here, the total DNA is taken, in which two major approaches are there. One is expressed sequence tags. Second one is sequence annotation. Expressed sequence tags. Here, all genes are identified that are expressed as RNA. That means you will take the genome, in which where the gene is present, they express out as RNA. These are taken outside. With the help of RNA polymerase, you can go for that. That's why genes are identified. Many geneticists followed this major approach. The rest of the members, they followed the sequence annotation. This is sequence annotation. Here, the total whole genome is taken and separated into coding DNA, non-coding. Coding sequence, it has functions. Non-coding sequence, no function at all. Finally, both are assigning different regions of the sequence with functions. So with the help of sequence, expressive sequence tags, genes are identified. With the help of sequence annotation, whole genome is sequenced here. Here the genes coding and non-coding. Finally, assigning them to the regions of sequence with functions. So that's about this area. Now see. The another challenge, actually it's a challenging project here. Another challenge task was assigning the genetic and physical maps on the genome. Genetic map and physical maps are there, and assigning that genetic map and physical maps are on the chromosome. It's very, very challenging task here. Then now the salient features of human genome. At the end of the project, the geneticists who are involved and they came out with their observations here. Those are salient observations. Human genome contains 3,164.7 million base pairs. It's an exact number. 3,164.7 million base pairs. Then, second observation, the average gene consists of 3,000 bases, but length varies always. So, largest known human gene, and whose length is 2.4 million bases, which is encoding dystrophin. 3,164.7 million base pairs. That is the exact number. Then they measure the length of the gene. The average length of the gene is 3,000 bases. Be careful. Gene 3,000 bases, not base pairs. Then the largest known gene that is encoding dystrophin is a muscle protein, 2.4 million bases. Then after that, the total number of genes is estimated as 30,000. 30,000, and actually previous estimate 80,000 to 1,40,000, but they identified 30,000 here. And these genes, when now seen them, and person to person, what is the identicality? 99.9%. 99.9%. It is amazing. Because people look different with different ideas, different morphology, different attitudes. But the DNA, 99.9% is identical here. Actually 30,000 genes are identified, in which 50% genes functions are not known. Unknown for 50% of the discovered genes. Suppose it is 30,000, 15,000 genes are not known. Functions, that's it. Less than 2% the genome codes for this. So that means total 3,164.7 million base pairs. Average length of the gene is 3,000 bases. Longest known gene, largest known gene, 2.4 million bases encoding dystrophin. And the protein coding genes are 30,000, in which 50% genes functions are unknown. Total protein coding genes in our total genome, it is less than just 2%. This is the 2% here. Less than 2%. Then the vital area, repeated sequences this is. Repeated sequences make up very large portion. Simply they are asking which of the following is the major portion of human genome. Repeated sequences, they make up very large portion. What are these? They are not genes. They have no coding function. See this. They have no coding function. But one point is present here. They shed light on chromosome structure, dynamics and evolution. So in our genome, the major portion is repetitive sequences. They have no direct coding function. They are not genes. But they shed light on chromosome structure, dynamics and evolution. Now this is a memory-based area. The chromosome that was sequenced last, the first chromosome, it has 2,968 genes. Highest number of genes are present on chromosome number 1. Least number, fewest, 231 Y chromosome. The latter, scientists have identified about 1.4 million locations where single-base SNPs, polymorphs. These are the applications here, normal applications only, but this is most important, you see this. Actually, 30,000 protein coding genes are there. Every protein coding gene is producing a protein. The protein undergoes post-translation modification. It will become an enzyme. That enzyme conducts one biochemical reaction. That means in our body, 30,000 biochemical reactions are happening. All reactions occur at a time. But understanding them, influence of one reaction on the other reaction, we cannot estimate, but now it is possible. Thousands of genes and proteins work together to orchestrate the chemistry of life. Since the beginning of the video, you are observing my mannerisms, my expressions, my movements here, that chemistry of my life was orchestrated by all 30,000 genes here. At a time you can study. That's about Human Genome Project. Second subtopic, DNA fingerprinting, final subtopic here. Here in this DNA fingerprinting, it is the very quick way to understand to compare the DNA sequence of any two individuals. Any two individuals are there, you wanted to compare them. You wanted to establish the biological relation between the two members. It's a very quick way, very important. Then after that, here, when you have seen the repetitive DNA, just now we explained, the major part of our human genome is repetitive DNA only. So DNA fingerprint is involved in some specific regions of DNA sequence called repetitive DNA, and you see that. Here, how to identify them? Very simple. Very simple. Actually, 90 out of 3 billion nucleotide base pairs, 99.9% is identical. Leave it. 0.1% we showing variation. That variable DNA is our genetic markers. How to identify them? Simply we will take the chromosome. We will remove the proteinaceous pellicle that is covering the chromosome. We will take the DNA outside. We will uncoil it. We will conduct a density gradient centrifugation. During that time, some major peaks will come out. The major peaks consist of bulk DNA. Bulk DNA means identical DNA. That 99.9% common for you and me, that is present in major peaks. Bulk DNA, we will remove it. Minor peaks, where you can see, that is satellite. Mini satellite, this is a point here, very important. In which genetic markers are there? What is genetic marker? Any molecular or physical characteristic that differs from person to person. Any physical or molecular characteristic that differs among individuals that can be identified easily in the laboratory is called as a genetic marker. They are present in mini satellites, just 0.1%. So, major peaks, bulk DNA, 99.9, remove it. It is common for every person. 0.1% is variable, that is mini satellites you can see in this area. At the same time, and these are showing inheritance from parents to the children, and those are polymorphs. That is DNA polymorphism you can see here, and the fingerprinting is the basis of paternity testing. Some medical legal cases are present here. In that condition, who is the actual biological father of the child? That is paternity testing. In that condition, this is ultimate. With the help of blood groups, you can discover he is the father, he is not the father. We can say like that. But there we cannot go exactly. But here you can go exactly. Why? Because paternity testing in the case of disputes. Why? Because the father's DNA is identified. Then polymorphism, very important. Poly means many, morphism means morphological forms. Here you can see the new mutations may arise in the individual either in somatic cells or in germ cells. Mostly they occur in the junk DNA, non-functional. 95% of our DNA is non-functional. In that junk DNA, mutations occurs here, both in somatic cells and germ cells here. Somatic cells will carry from one generation to other generation, no problem at all. But whereas germ cells, we see this here. Germ cells. See this line, very important. If a germ cell mutation does not seriously impairs individual ability to have offspring, we can transmit. Somatic cells, that will happen, but the germ cell is the condition here, and that's the situation. Then after that, come to this area. So through the sexual reproduction it happens here. Now the definitions are given for DNA polymorphism, very, very important. See, if more than one variant at a locus, variant means allele. If more than one allele at a locus occurs in human population with a frequency greater than 0.01, there is a locus. In that locus, one allele is there and showing a frequency greater than 0.01. 0.01 means 1 in 100. That is a DNA polymorphism, that's it. Then, this is another important definition. If an inheritable mutation is observed in a population at high frequency is referred to as a DNA polymorphism. So, greater than 0.01, that's one important point. It is inheritable, most important, see this. It should show inheritance. And that is very important. It should have high frequency, that's it. Then the DNA fingerprinting technique was initially developed by Lac Operon. He's a Great Britain. This man belongs to Great Britain. While he was working with the gene of myoglobin, he noticed that there is a change in person to person. And these are variable number tandem repeats. The genetic markers are variable number tandem repeats. Tandem repeats means there is a fragment of DNA with a particular length, with a number. It is repeatedly repeated. Tandem is repeated, repeatedly repeated. These are the genetic markers. Now, how to take these genetic markers outside? How to go for the protocol of DNA fingerprinting? First of all, isolation of DNA. And a cell is given to the person, and from which the nucleus is isolated, nuclear membrane is removed, chromosomes are isolated, chromosomal proteinaceous pellicle is removed, the total DNA is isolated. The first one. Isolation of DNA, isolation of DNA from this area. Okay. So after that, we have to come to the next one, that is digestion of DNA by restriction endonucleases. First of all, from the cell, DNA is isolated. After that, digestion of DNA, that means by adding restriction endonucleases, DNA is broken into pieces. It is also called fragmentation. After that, separation of DNA, fragments by gel electrophoresis. Based on the sizes, these are separated. First, we will take the DNA outside. Then after that, we will break the DNA into pieces by adding restriction endonucleases. Then we will separate the DNA pieces based on the sizes. Small pieces can travel longer and long pieces can travel. That's it. Then after that, these are the DNA pieces are present, which are transferred onto the nylon membrane. That is blotting method. Southern blotting method developed by E.M. Southern. Then hybridization. Later, a radioactive probe is taken and it is added to this and VNTR, that's it. Then detection of hybridization, DNA fragments. First, we will take the DNA outside. Later, we will break the DNA into pieces. We will separate the DNA pieces by using gel electrophoresis. After the pieces are transferred onto the nylon membrane. And after that, we are using a radioactive probe that will bind with VNTR. And finally, with the help of autoradiography, which are identified. Now, VNTR belongs to a class of satellite DNA. Present in minor peaks here. The size of VNTR varies from 0.1 to 20 kb and that's it. Then it differs from individual to individual in a population except in the case of monozygotic twins. Very important. Monozygotic twins means they are coming from a single zygote. So that is not possible, that's it. Then application of forensic science, it has much wider applications such as, and these are the conditions here, and that's it. Now after that, see this area, this diagram is given, NCERT only. Here paternal means chromosome from father, maternal from mother, and this is individual A, individual B, and both are taken. And here you see, this is the DNA obtained from crime scene. One crime happened in your residential area in which the forensic experts have taken a sample from which they made a DNA fingerprint. This is a DNA from the crime scene. Then later, these two are suspect persons, A and B. And simply they match this. Match this here, you see the B is matching. B is matching with this. Not with A. Individual B is matching. And you see the height here. Height indicates the length of the fragment. And this is the condition here. So, DNA from the crime scene matches with individual B and not with A. That's it. So, that's about condition. Now, here another important point, determining population genetic diversity is also. Actually, this DNA fingerprinting is useful in forensic science, medical legal cases, then pedigree analysis, then identification of distance of one animal to other animal, anthropological studies, etc., etc. Okay. So after that, I will show you some important questions. Before that, we will have a small video, and after that, you will see the 2 reels, and after that, we can have some model questions. That's our NCERT. Yes, look at the screen. Here I am showing some of the questions here, model questions. And here, see, pick out the incorrect statement from the following. 4 statements are given. What is incorrect? The total number of genes is estimated to 30,000. Nearly 1.4 million locations are with single nucleotide base differences. Repetitive sequences are thought to have no direct connection, and nearly 3% of the genome codes for protein. Wrong. It is less than 2%. Less than 2%. Now, coming to the second question. Which of the following is not required for the technique of DNA fingerprinting? That is identification of genes that are expressed as RNA. That is expressed sequence tags. And it has no business with our DNA fingerprinting. Then coming to the third one, the longest known gene or the largest known gene of human codes, and it codes the muscle protein dystrophin. Muscle protein dystrophin. Then fourth one, which of the following was a major partner during the early years of Human Genome Project? That is Wellcome Trust of UK. Wellcome Trust of UK, he was the major partner in the early years of Human Genome Project. Here I am showing one 2-column matching. Here human genome and 3,164.7 million base pairs. Chromosome 1, 2,968. Then SNPs, 1.4 million locations. Y chromosome, 231. You have to match it. It was also one kind of question. The next question, automated DNA sequencer work on the principle of a method developed by Frederick Sanger. Frederick Sanger is known for that. Automated DNA sequencing method, and that work on the principle developed by this man here. Then small peaks that are formed during the density gradient centrifugation of DNA are referred to as. While you have isolated DNA, we are making a density gradient centrifugation, the major peaks and minor peaks. The minor peaks are small peaks. Major peaks are having identical DNA. Minor peaks, a small amount of DNA, and where you can see satellite DNA. There you can have genetic markers. VNTR, variable number, tandem repeats. Then after that, question number 8. DNA polymorphism is referred to as inheritable mutation in a population at high frequency. 2 components are required, and it should show inheritance, very important, and it should have high frequency. High frequency, these are the 2 components. Be careful. Every time they are mixing these 2, it will show inheritance. First point, it should have high frequency. Then human genome fair question, direct question, memory based, numerical. That is Human Genome Project approximately consists of 3 into 10 to the power of 9. And after that, the 10th question, in the technique of DNA fingerprinting, autoradiography is used for it. Is the final step. When the radioactive probe linked with the VNTR, finally we will detection of hybridized DNA fragments with the help of autoradiography. When the VNTR is hybridized with the probe and with the help of with the autoradiography, and we are detecting hybridized DNA fragment, that is genetic marker. Then DNA fingerprint differs from individual to individual in a population except in the case of very important monozygotic. Monozygotic twins, these are identical twins. The two babies are coming from a single zygote. Their DNA fingerprint is exactly same. Except in this case, in any case, we can trace a person except monozygotic. Then coming to the 12th question, what percentage of genes functions are not known out of the total? 50%. 50% genes, 30,000 protein coding genes, in which just we know the functions of 50%, generally 15,000 genes, rest of the genes functions are not known. Then which of the following makes a very large portion of human genome? Repeated sequences. The repeated sequences are making large portion. They have no direct coding function. They shed the light on chromosome structure, function and evolution. Then 14th question, Human Genome Project was closely associated with the rapid development of a new biology, the biology area, that is bioinformatics. In the first paragraph, final line, that only. So that means it is a link to bioinformatics. And the final question, the technique of DNA fingerprinting was initially developed by, that is, Lac Jeffries, and who identified the differences of DNA while he was working with the genome myoglobin. So these are some of the model questions I am showing. Don't think that these are the only questions. There is a possibility of designing any model, any question, even assertion reason also, and multiple statements also, matching also, different kinds are present here. Just I am creating an idea. So go through all the points here. Definitely you will consolidate this topic perfectly. Definitely you will reach the answer to every question which you are designed in this area. So go in this manner. This is the purpose of the chapter at a glance. Make use of that and be perfect and go. The preparation is smart. So I wish you all the best. God bless you.